to come and give your departmental seminar. Like I said earlier, there's sugar up here for anybody who wants some energy, and I'm going to have to try and be coordinated, so if you see me pointing the microphone thing, it's because I have to figure out to use the other hand. So I wanted to share some research with you today that we've been doing in Lincoln for quite a long time. I've been there for 10 years, but the studies that I'm going to present were established long before I got there. So very lucky to come in on some well-established studies and kind of build on what had already been done there over time. I'd like to acknowledge my co-authors here, Marty Schmier, Kathy Stewart, Aaron Sindelar, Candace Williams, Gary Varvel, Rob Mitchell, and Brian Weinhold, who all in one way or another contributed to these studies. So before I get started on the research part, I've got my little spiel on talking about the USDA Agricultural Research Service. Prior to my joining them as a postdoc 12 years ago, I had no idea that they existed. And so every time I give a seminar, I try to talk a little bit about USDA and what the Agricultural Research Service does. Not so much of a big deal here since you guys are a land grant university, and so you may have more interactions with them than other universities would. The USDA is the largest civilian department in the US government, and we have several different mission areas. The strategies for the USDA, I'm not going to read all those out to you, but they change with every administration, and in Lincoln, Nebraska, the two that we work with in my unit are really to look at the stewardship of private lands and also ensuring food safety with the work that we do. The research in the USDA is primarily conducted by the Agricultural Research Service, but we do have a smaller amount of research that's conducted by the Economic Research Service, the National Agricultural Statistics Service, and NIFA, which y'all probably all know because you've applied for grants or something at one point or another. Again, the priorities change with every new administration. So the Agricultural Research Service itself is one of about 17 different agencies or offices within USDA. We're all administratively separate. We have over 690 peer-reviewed projects among 15 different research programs, so we split ourselves up by discipline in that way. We've got about 2,000 scientists and 6,000 other employees that support us. And 90 plus research locations, and they're not all in the states. Many of these that are out of the states are biocontrol locations because we have a lot of pests that we've eliminated here, and we certainly don't want to study those pests here and reintroduce them when they're not a problem. So we go to countries where those issues are still existing. In general, the various studies that have been done on federally funded agricultural research is that for every dollar that's spent of taxpayers' money, we return $1.35. So a 35% return on investment is pretty darn good when we consider people play the stock market and they think a 10 to 12% rate is very, very high. So for a small agency, we give a lot of bang for our buck. We've had a lot of major technology transfer accomplishments, and because we're a small agency and we don't do public service announcements, I just wanted to brag on us for a little bit. So a lot of the work that's been done on plant diseases and resistance, a lot of that comes out of our research programs, which we then transfer those technologies to private industry so that they can contribute to the economy. We've also been involved in freezing fruits and vegetables. So a lot of that's in your grocery cart. We've got a very active program on citrus and citrus greening. That's in Florida. And for those of you, I'm very sorry to say that reduced fat cheese, that came out of our labs too. Catfish aquaculture, that was something that was primarily developed by us and again passed on to industry. Lactaid, for those who are lactose intolerant, we contributed to that as well. The commercialization of poinsettias in the US, that was developed by our labs also. If you're wearing a cotton t-shirt today or something, you'll know that maybe 10 years ago, if you threw something cotton-based into the dryer, that it would shrink up. And now we have cotton fibers that don't do that. They're wrinkle-free and shrink-free. We had something to do with that as well. We've also got one of the biggest patents based on cornstarch. It's an absorbent called Super Slurper. it's just disgusting sounding, but it actually absorbs more than 2,000 times its own mass in water. So you'll often find it in wound dressings, diapers, other things like that. DEET, yes, we came up with the development of that. And we also had something to do with the finding the strain of penicillin that was used in World War II and the widespread commercialization of that drug, which saved a lot of lives. So to get back on track now, I was going to talk a little bit about our bioenergy research. So bio-based energy, this is really an old idea. If we look at how energy consumption in the U.S. looks over time, for like forever, it was really a lot of it's biologically based. And it was, even though it was once a major energy source in the U.S. and we've since developed and gone beyond that, bio-based energy sources continue to be the dominant energy source for other parts of the world. So if we look at the numbers here, in 2014, the energy consumption was that we're looking at a total consumption of 98 quadrillion BTUs. That's like humongous, of which 10% come from renewable energy. These numbers may have changed because these numbers are about 5 years old now. But of that 10% renewable energy, 50% of that is expected to come from biomass. And there's just a little, what is a BTU? It's the amount of heat required to raise the temperature of 1 pound of water by 1 degree Fahrenheit. So for example, 1 gallon of gas has over 120,000 BTUs in it. And 1 quadrillion BTUs is 10 to the 15th that. So it's a lot of energy. If we think about biomass feedstocks in the United States, we have a few different sources to consider. Forest-based products, and if we look over time, more and more over time is expected to contribute to that feedstock. We have agriculturally based feedstocks, which might be crop residues. And then we have energy crops, perennial grasses, short rotation woody crops. And if I just focus here on the stover, I'm going to talk more than just stover here. The stover supply is estimated to be anywhere between 170 to 260 million dry tons per year. And the energy crop supply is even bigger than that, but it's a lot more variable because it's still something that's in development. So that's a really big number. And just to give you some idea for scale, how much biomass is that anyway? So let's assume that 1 round bale is about half a dry metric ton. And just as a round number, let's look at 200 million dry tons a year. That's 400 million bales of stover every year. That's still a really big number. So I'm looking at you guys' football stadium, and if I calculate the volume of the bowl in your stadium, we would be filling that 600 times every year. And that's just for stover, not including the grasses or the forest-based products in this calculation. So we're talking about a lot of biomass every year. That's a lot of biomass every year. So, when we think about biomass production, it's not just solely going for bioenergy production. There's a lot of other uses for it, and it's really part of a larger system where we have not just one product off of our row crop systems, we have grain and biomass. In Nebraska particularly, we have a very, very active livestock industry. We're the #1 red meat producers in the U.S. You'd think it was Texas, but it's not. We've got a lot of grasslands and grazing, and then we also have a lot of ethanol production. Not as much as you guys, but we have a lot of it too. We're #3. If we think about the Renewable Fuel Standards that was established by the Energy Independent Security Act in 2007, there are a number of targets that act established for looking at renewable fuels and what counts as a renewable fuel and how that translates to greenhouse gas reductions. So if you're any kind of starch-based biofuel, the expectation is that you're going to have 20% less system emissions, greenhouse gas basis, than petroleum-based fuels. If you're in advanced biofuels, you're going to reduce that by 50%. So that would include non-starch-based biofuel, non-corn starch-based biofuel, so sorghum, wheat, sugarcane. And if you're a cellulosic or other ag waste-based biofuel, the expectation is that you're going to have 60% less greenhouse gas emissions. And that would be fuels derived from cellulose, hemicellulose, lignin. So what counts as a greenhouse gas? We're normally concerned more with nitrogen as a trace gas because it's very reactive, but we also have concerns with methane production. Maybe you've heard some of this in the news recently. There's always a lot that talk about cows farting or whatever, but it's really not their farts, it's their burps that the methane is coming from. And we can handle some of those by changing feed. In ag systems, we have ammonia, nitrous oxide, and methane, but just to clarify, ammonia is not considered an atmospheric greenhouse gas. In order to be a greenhouse gas, you've got to have a lifetime in the atmosphere that is long and has a temperature forcing potential. In the case of ammonia is not a gas because it's highly reactive. It's gone within 24 hours of being emitted. So I'm not going to talk about that gas. Nitrous oxide is the same as laughing gas. You may have had it if you go to your dentist. I like to think that's why my students feel so good when they go gas sampling for me. But it's not, there's not that much. But it is an atmospheric greenhouse gas. It's got a lifetime of 114 years in the atmosphere, and it's got 300 times the temperature forcing potential on a 100-year time horizon compared to a molecule of CO2. Methane, also a greenhouse gas, a 12-year lifetime, and about 20 times the temperature forcing potential of CO2. So I'm pretty much going to focus on just nitrous and methane here. Okay, what about CO2? Everything is in CO2 equivalents anyway. Why aren't we thinking about CO2 as a gas? In general, CO2 is in fact the largest source of gases that are being emitted, over 70% from the soils particularly. But it's so variable because it's based on biological activity that in ecosystems we can best look at carbon changes not by looking at CO2 emissions, unless you're lucky enough to have an eddy flux tower set up. But for the rest of us that don't have that kind of money, we just go soil coring. So we're going to look at net changes in soil organic carbon over time as a way of looking at those carbon-based changes. So in our unit, what we typically do is we'll surface soil sample every 3 to 5 years. So that might be down to 30 centimeters. But then every 10 years, we'll go down 5 feet. So over 2 meters, over a meter. In the United States, agriculture is contributing 9 to 13% of the total emissions. That number's actually been going down from year to year because other sectors are emitting more. And if we look at 2016, both the combination of nitrous oxide and methane contributed to 16% of the total U.S. greenhouse gases coming from agriculture. But agriculture is the big contributor for nitrous oxide emissions, more than 80% and more than a third of the methane. And the nitrous oxide sources are pretty much have to do with our fertilizer and soil management. And the methane's coming, most of that's coming from enteric fermentation. So how do we define soil greenhouse gas emissions? These are what we call direct emissions. They're emissions of nitrous oxide that's coming from the soil due to microbial activities that are occurring in the soils involved with nitrogen transformations. There are abiotic pathways for nitrous oxide production. Those tend to be minor, but they're there. I just want to acknowledge that for any physicochemical people out there that don't want to be pushed aside. Again, global warming potential of 1 molecule of CO2 for a 100-year time horizon, we'll call that 1. So then methane has 21 times that, and nitrous has 310 times that. Those numbers tend to fluctuate depending on which IPCC report you're looking at, but it's within that same range. And again, just kind of concentrating on methane and nitrous here in this talk. Where does agricultural management come in? How do crop type, and specific management practices affect biomass production, but more so the global warming potential of these production systems. So if we think about a system type, are we talking about perennial crops or annual row crops? What are our fertilizer management like? What's our tillage like? Do we have any kind of biomass removal occurring? How much of it are we taking off? How frequently are we taking it off? When are we taking it off? And to calculate global warming potential of the system, and we're going to do this in CO2 equivalents so we get all of our numbers on the same basis, what do we need to know? We need to know all of our non-CO2 soil greenhouse gas emissions. We need to know changes in soil organic carbon stocks, because now we're covering N, methane, and carbon, CO2. The total energy usage for the management that's involved, so that would include diesel or electricity used to run machines, irrigation, what have you. And so basically your global warming potential is any gas emissions that you have, plus the energy use that you use, that's all gas that you're emitting, minus the storage for carbon that you're getting in the soil. My caveat here is that I'm going to present to you global warming potential of just the agricultural production component of the larger system. I'm not going to include transportation costs. I'm not looking at the whole field farm gate to whatever the larger life cycle assessment. This is just for the agronomic production phase of these crops. So how do we quantify direct greenhouse gas emissions from soils? One, you need a small army of undergraduates to go out and take the measurements, and a great technician who keeps them all together. We use static chamber measurements. Weekly during the growing season, we get measurements. In the early or late season, before the plants are really going or when they're starting to slow down, we'll go every 2 weeks. And then in the non-growing season, we'll go every month to get baseline numbers. And yes, when it snows, we still go out there and we'll get numbers. And we do this with these static chambers that are based on the buffet pans. They're stainless steel. They're lightweight. They are economical. And so a very good use of taxpayer money to get at some high-powered data collection here. And in addition to taking gas samples, we will get some ancillary data, including air and soil temperatures and volumetric water content at 10 centimeters depth. When we quantify total fuel usage, I went back and I got some of these assumed numbers based on a publication from Paul Adler. And this is going to depend on the equipment type and size, the number of passes of that equipment, the operation type and intensity. So you can imagine if you have a tillage versus a no-till system, there's a lot more fuel being used there. Carbon intensity, we converted that using the California Air Resources Board conversion. And so here's some example values for what the equivalent CO2 emissions are for these various field operations. In the big scheme of things, those numbers are really pretty low, just for relative purposes. So biofuel feedstock systems. Which systems are we going to look at today to look at what those global warming potentials are? We have two potential systems that we're really focusing on hard as potential biofuel feedstock production systems. One is high-intensity systems, such as irrigated continuous corn. We got a lot of that in Nebraska. The other potential place where we can get biomass is on these marginally productive lands. And there's a bit of a competition there occurring between annual crops being grown on these marginally productive lands, which probably aren't that profitable because they're marginally productive, versus perennial crops that you can grow on the same ground if we would only give producers a potential revenue stream for doing that practice, which we still are in the process of developing that. So let's look first at this irrigated continuous corn system. So we established this in Ithaca, Nebraska. This is about a half-hour northeast of Lincoln. The University of Nebraska maintains a very large research farm out there, and this is on that farm. And what we have here is basically a study that was established in 2002. And it has 6 reps in it, but we only measured gases out of 4 because we measure gases out of a number of different studies, and I have to avoid mutiny in my group, so we do a selected number of samples and replicates. And in this study, we look at no-till systems versus conventional tillage, which is a disc till. We have 3 levels of corn stover removal where we take none of it off. We take a moderate amount, which is every 2 rows. So you can actually see, I love Google Earth. This is amazing, right? So you can see where we take 2 rows off, every other 2 rows, and then the next year we'll flip. And so we'll take the 2 rows that we didn't take this year, we'll take next year. So that goes every year. That's how we do that. And then the full residue removal where we take off as much as mechanically possible. So the greenhouse gases that we measured, at least that I'm presenting here, was just on a subset. I'm only going to show data for till versus no-till. And then for no stover removal versus the full, the maximum residue removal. And that's just as a way to kind of bracket the most extreme treatment that we have. And what I'll show here in changes of soil organic carbon, this is going to be on a fixed mass basis, which is important when you have tillage comparisons because soil density is going to be quite different. In a tilled system that's highly disturbed, it's a lot fluffier compared to a no-till system where it's condensed and it's consolidated. So this is on a fixed mass basis, which roughly corresponds to the top foot of soils. And we did 3 measurements of this, one in 2001, 2010, and 2014. So that's what it looks like when you leave all the residue on the ground. And we take that residue off with a flail chopper. It blows the stover into a wagon that we then weigh the amount of stover that's being removed. And there's, with all the stover, that's the maximum amount of stover being removed. So you can see there's still quite a bit of it on the ground. The process of taking it off kind of breaks up the stover, so there's still some there. It doesn't always stay there because Nebraska's really freaking windy. And so by the end of winter, a lot of that's gone. But that's what it looks like immediately after residue removal. So our mean high residue removal rate is very high. We don't recommend this as something that a producer should do, but as a experimental treatment, again, to kind of bracket the extremes of management, it's about a 75% removal rate. Our irrigation inputs here, we let the farm manager manage it just like anybody else that's producing out there. So it's anywhere between 2.5 to 16.6 centimeters per year, just kind of depended. That 16.6, that was the 2012 drought year. It was really pretty bad that year. That's unusual. And so what I'll show here is gases that we collected between 2011 and 2015, so over about 100 different sampling events. So if we look at the daily emissions occurring off of those measurements for each of these 2 gases of interest, and you're going to see this table again for another study site. Over 1,500 individual measurements. The flux rates were very high at some times, but on average, you know, the median rate was pretty low. The mean, because these maxes were really driving that average higher, is around 14 and a half. What we also determined was that of all of the measurements that we took, 84% of all those individual measurements were actually non-zero fluxes. So 16%, there was some number that we could calculate, but it wasn't significantly different from 0. So 84% of those were significant fluxes, of which 2% was consumption. Nitrous oxide was not blowing off, it was being sucked into the soil. So very low percentage, but that still happens. It's a biological system. Methane, again, a little over 1,500 individual measurements, very low fluxes. Again, 84% of them were non-zero fluxes, but 44% of those measurements that we took were consumption. And this frequently happens with methane. Methane production is an anaerobic process. In our upland, well-drained row crop systems, the reverse happens. We don't get methanogenesis, we get consumption of methane occurring. So a lot of the methane gets pulled out of the atmosphere and is being consumed by methanotrophs in the soil. Generally speaking, methane is negligible compared to nitrous oxide emissions. But we're going to include it just for completeness sake here. Peak nitrogen oxide fluxes occurred within 1 to 3 weeks after fertilization. We always increase our frequency of sampling after tillage events or fertilizer events because we want to try and capture these peaks. And what we find is what the literature tells us that we're going to find. You get the highest fluxes occurring when your soil temperatures are warm and when they're wet. So it's nice to see that borne out by the actual data. Methane fluxes, they weren't correlated to soil temperature or water. They were just so low, they were so variable, there's just not a whole lot going on there. So if we look at the total direct nitrous oxide emissions or the soil emissions cumulatively over those years, what we'll find is that there was a lot more variability from year to year in the disk system. So the more disturbed that system is, the more variable those nitrous oxide emissions were during that 5-year period of time. If we look at just the stover removal, we see that there's a significant amount of more nitrous fluxes from where we don't take the stover off. We've kept the stover on, we've given the microbes a food source, we've protected the soils, we've probably made the soils a little bit more moist, the conditions are better, so they're doing more. And we didn't see any interactions in treatment here. What about carbon stocks? Again, no interactions between the management systems. Tillage management, the rate of soil organic carbon change in that 10-year period of time, we did see losses. The losses were not significantly different from zero for the no-till system, but they were significantly different from zero in the tilled system. When we took stover off, we really exacerbated that SOC loss. So again, here, you don't take any of that off. It still looks like you've got a reduction in soil organic carbon, but that's not significantly different from zero. But then as soon as you take that stover off, that is significantly different from zero, and it's significantly different than if you'd left that residue on. That's a 9% loss. So, kind of in order, what we see is if you have high residue removal rates, you tend to see the most SOC loss, and then tillage disturbance, that's the next highest. So we put all that together with all of the fuel usage and energy usage that I mentioned earlier, and we look at what is the ag phase global warming potential of these systems when we combine fuel use, soil organic carbon change, and soil greenhouse gas emissions. Again, no management interactions. If we look at tillage management, there's a marginal increase in the global warming potential for disc tillage compared to no-till. A little bit higher. Stover removal definitely increased global warming potential in that system. That's also an extra machine pass that you're taking there to bale, chop it up, take it off, bale, or whatever you're doing. But the bottom line is that in this irrigated production system, it didn't matter what management you used, they were all greenhouse gas sources, all of them. Which is kind of depressing because you want to think that we could take some of the residue off of those systems as a potential for a biomass source. Excuse me. So in general, conservation practices, leaving residue on, using no-till, those things did decrease the global warming potential, but it didn't change the fact that the system itself was a greenhouse gas source. Stover removal increased the global warming potential because the soil organic carbon losses were really high, and those offset the decreases in soil greenhouse gas emissions. Remember, when we took stover off, greenhouse gas emissions went down, but we lost so much soil organic carbon, that offset was just too great. What we did find was that pairing those conservation practices, because often people will say, hey, if you're going to take stover off, you need to go to a no-till system. What we found is that there was no additional benefit of pairing those two practices. Each one individually, they did reduce the global warming potential, but using them together didn't have an additive or synergistic effect in further reducing the global warming potential. Nonetheless, there are other reasons why a producer might choose to do both of those companion practices together. Some of these deal with soil health and erosion concerns, so it's not just going to be, I don't know of any farmer that's going to just say, hey, my global warming potential is a reason for me to make a certain management decision. That's really not the way that's going to go. So we covered that high intensity irrigated continuous corn system. Now let's go way to the opposite side. We're going to go to marginally productive land where we're looking at perennial versus an annual crop, side by side. What makes it marginally productive? We pick these sites based on the fact that the long-term average yield production at this site is less than 75% of the county average. That's our definition for marginal. Now, one of the reasons why this site is marginal is because the Platte River is rather close by, and this has got a sandy lens of river sands, sometimes as close as 30 centimeters, 35, 40 centimeters from the surface. So there are some really, really dry areas here at this site. So this site was established in 1998. Same spot is just up the hill from that irrigated continuous corn study. 3 field replicates where we're looking at no-till continuous corn, switchgrass, and two different cultivars of switchgrass, Cave and Rock and Liberty. Liberty was a biomass grass that was developed at Lincoln by Ken Vogel and Rob Mitchell, and it's bred to be huge. We had 3 different levels of nitrogen application here. And we had a split treatment in which we looked at the effective harvest timing. Again, I only picked a subset of these to share with you. So we're going to look at post-killing frost, which is harvesting the grasses after frost. So it's maximum biomass. You've also given the plants a chance to kind of translocate some of those nutrients back down to their rhizomes to overwinter. And then we're going to look at 50% stover removal, because those are the 2 scenarios in which we're going to be removing biomass. Post-frost, post-killing frost for the perennial grass versus taking off half of the stover if you were in a row crop. So here's that table again. And what you'll notice in this case, we had over 2,600 individual observations. And a little bit different because we're in a different environment. Only 69% of the observations were actual non-zero fluxes. Same, very low percentage of those were actual consumption that occurred. And methane fluxes, again, very low. 56% of the non-zero fluxes were consumption. So even more consumption occurring here in this marginal site than compared to that irrigated site that I just talked about. So we had 94 different sampling events here. And again, just like the irrigated site, we see peak nitrous fluxes occurring 1 to 3 weeks after fertilization, when soils are warmer and when soils are moister. That all makes good sense. We did have one sampling date where the methane fluxes were incredibly high. And that just goes to show that there's a lot of soil heterogeneity and things that are happening on a micro level. Sometimes we catch those events and sometimes we don't. Unless you have a continuous system like an eddy flux tower to capture all of those fluxes, we don't have an expectation that we're going to catch them all when we do the static chamber measurements. We just hope that the frequency at which we take the measurements is adequate, at least to represent the relative treatment effects on the gas fluxes. So if we look at the 3 different nitrogen fertilizer rates, and again, we're looking at the cumulative annual emissions that are occurring. They're pretty low when you don't add any nitrogen, but they're still occurring because it's a biological process and there's still native nitrogen that's being mineralized out of the soil. You add a little bit of nitrogen and then your nitrous fluxes get a little bit higher. You add twice as much of that again, and you see a lot more fluxes occurring, and there's a lot of interannual variability, regardless of which treatment that you're looking at. If we summarize those into the 5-year means, Again, low fluxes occurring when you don't add any nitrogen, almost twice as much when you add 60 kilograms of N, and then even larger, that's more than twice as much when you add 120 N, because at some point, for a certain optimum nitrogen rate, if the plants are not going to utilize that, all of that nitrogen that you add, whatever's left over, is susceptible to loss. So what does this tell us? That this 120 N is probably above the optimal nitrogen rate for these two crops at this marginal site. Nonetheless, if we go back and calculate what is the emission factor, so we're going to subtract out the native emissions occurring at 0 N to figure out what percent of the nitrogen that you added is being blown off as nitrous oxide. IPCC tells us they use like a flat 1% expectation. What we find is that, yeah, that's pretty close, but there's some variation there depending on management. And it's certainly, we find it's way above that for when you add too much nitrogen. So using that flat 1% rate, yeah, it's pretty good for doing back the envelope calculations and really broad generalizations, but there's a lot of other things that happen in the field that can change that number from year to year. What happened to the carbon in these soils? So if we look at these three different systems, and I'm going to refer to our cave and rock system and our Liberty system as cave and rock being continuous switchgrass, because that's been there since 1998. I'm going to talk about Liberty as being a rotational switchgrass system, because we actually started off in 98 with a different cultivar, Trailblazer. Trailblazer totally croaked after 8 years. And had we been in a situation where we could not have funded that study longer than that, we would never have known that. So what we did was, after Trailblazer choked out, we replaced it with 2 years of soybean, and then we went back in with this Bioenergy Liberty cultivar. So we think that this rotational switchgrass system simulates what producers would probably do on their own farms. Every 10 years or so, you're going to go with the next best cultivar, and you're going to replace the one that you have with a more vigorous cultivar that's available to you. So in general, what did we find? On this marginal site, which before had been corn, soybean, and tilled every year, when we went to no-till for corn or put perennial grasses in them, all the systems gained carbon. Some people would have expected to see carbon losses because we still had row crops up there, but the adoption of no-till, even on that marginal site, that led to carbon gains in corn that in some cases weren't all that different than what we were seeing under switchgrass. That was kind of surprising to us. We particularly see this in corn when we keep the stover on, that's not surprising either. That's very similar to what I showed you for the irrigated study. But for both switchgrass systems, all of these, except for maybe this guy right here, and he was new, all of these were gaining carbon. We had no differences in fertilizer rate, which that was kind of surprising. We would have thought that as you increase fertilizer amounts, that your biomass would increase, you'd get more carbon input into the soil. But the fact that we weren't seeing that also suggested that as you added fertilizer, you're also stimulating decomposition. So everybody seemed to be, whatever you were gaining in carbon, maybe you're blowing off a little bit more of it too when you give them more nitrogen. So, okay, the global warming potential, this is the big number that we're after for the ag production phase. How did no-till continuous corn with stover removal compare to these switchgrass systems? Well, if we look at no-till corn, what I'm showing you here is, here's the fuel usage in red, the direct emissions are in yellow, and the soil organic carbon change, that's in green. The net emissions coming off of this corn system, they were all positive, but not significantly different from zero. What does that mean? That corn system, whether or not you took stover off of it, was pretty much a greenhouse gas neutral system on that marginal site. If we look at the continuous switchgrass system, which is cave and rock, been there since 1998, we see a lot more going on here where we have a greenhouse gas sink or it's neutral. We have a lot more emissions occurring here in 120 N because this is a very mature switchgrass stand, and 120 kilograms of N is probably way overkill to be adding. So again, whatever the plants are not using, that's susceptible to loss. So we had quite a bit of nitrous oxide emissions occurring there. And then finally, if we look at that newer rotational switchgrass system, again, we're finding that they're either neutral or it's a greenhouse gas sink. That's not very surprising given that we have expectations for perennial grasses to be storing a lot of carbon and acting in this way to mitigate greenhouse gas emissions from the atmosphere. What was more surprising was the fact that we thought that all of the corn systems, like in the irrigated system, were going to be greenhouse gas sources. And they weren't in this case. They were neutral. So switchgrass global warming potential indicate that they're greenhouse gas sinks. No-till corn was neutral, but they could become sources over time. Just the kind of cumulative effect of those rates might not have been significantly different from zero if you annualize it. But the longer that system is in place, that's going to add up year after year after year. Soil organic carbon storage, that's the main driver determining whether or not your system is going to be a source or a sink. But it was really the nitrous oxide emissions that made that final determination whether you push that system over into whether it's storing carbon and mitigating greenhouse gases or if it's going to be an emitter. We think that a lot of the SOC change came from below ground inputs. The rooting systems of the switchgrass is really quite remarkable. But we had another study done that looked at, even though corn roots don't look as robust and they don't look as abundant as the switchgrass roots, they have a different way of sticking around. The way that the microbes use them and decompose them, they're still contributing quite a bit of carbon below ground as well, which we thought that was quite interesting. So the final thoughts. Perennial crop systems were greenhouse gas sinks. Yay. Annual systems were greenhouse gas neutral. And nitrogen management in this case, because they tended to be close to optimum, didn't really have an impact. Stover retention and annual cropping systems didn't affect the greenhouse source strength. It was still a source, but it didn't change how much. So what we think is that we're using, if we think about how we manage our farming landscapes, not just by field, but if you look at the landscape that we're growing food and energy on, if we can match certain management practices to a landscape zone type, then we'll probably be more effective at reducing the global warming potential of those different production systems. So the big question, can stover or switchgrass systems meet the mandated 60% reduction in greenhouse gas that the EISA 2007 had deemed that's what we should do? We don't know. I just showed you global warming potential for just the production phase of that system. To get at the big picture, we'd need to do a full life cycle analysis that would include the much larger manufacturing and transportation and all that stuff, which Marty is going to do. Okay, and my final thoughts. Long-term studies, they're super valuable. They're well equilibrated. As I said, I was very lucky to come in on these studies that were already established. And I get to sort of stand on the shoulders of giants here by using some of that data to do this analysis. Predicting system sustainability, it requires a lot of data. And unfortunately, most of the stakeholders who want to use that data don't have a lot of patience. So another reason why having the long-term studies are really valuable. There's a lot of spatial variability. There's a lot of temporal variability. And I think that's why we see the onset of a lot of these digital agriculture positions that are coming online, or agronomy departments that are now positioning themselves to be more digital ag. Sustainability outcomes are really, really variable. But I like to look at it like if you think of the doctor scale, when you go to the doctor, that the big weight, that's soil organic carbon change. That's kind of kind of estimate where you're at with how much you're gaining or losing. But that small little weight on the top that you move around, it's nitrous oxide. That's actually what's going to determine what's tipping the scale in one direction or another. So with that, we had a lot of university and industry partners. I did have multiple small armies of students and technicians that helped me with all of this. None of this work would be possible without them. And we had a couple different sources of funding here. So with that, I hope I didn't go too far over time or anything. I would be happy to take any questions. Yes, ma'am. Hi.
So, if I understood the take home from the last study, the REAP study, we should plant corn on marginal land?
No.
Improve global warming?
No. You know, I think what the studies are telling us is that whatever management decisions we make on certain kind of landscapes, we have a lot more flexibility to offer the producers by saying, okay, ideally we want you to go to something that is more perennial. Perennializing the landscapes in the long term, going to be more sustainable. But not everybody's going to make that decision because that doesn't pencil out for them. They can't pay their mortgage, they're not going to get their kids through school. Whatever their reasoning is, it's not in CRP anymore. They didn't have enough acreage to make it into a CRP program to get a payment. What we can offer them is saying, you know, at least for the time span in which we looked at these studies, even if you go to a row crop on a marginal site, it's relatively greenhouse gas neutral as long as you use a conservation practice. And so if that is what a producer chooses to do, until such time that there's a revenue stream for them by putting perennials on the landscape, then at least we can still push the idea of using a conservation practice as opposed to them just using a conventional practice on a marginal land. But by no means are we saying, yay, go plant up row crops on marginally productive lands. We're just trying to give more options for people. Right now, I'm not quite sure that bioenergy is exactly where all of our biomass is going to go. There's a lot of other alternative uses for it, and as those other uses come online, whether it's bedding or using the materials for some other source, like I think there's a couple businesses even in Iowa that are using the material for those erosion socks or something like that, and the industry's going really well. I mean, there are other possible places where the biomass can be used. But again, it's a really young industry and not many farmers know about it yet. So hopefully as that develops and we have more viable opportunities for to producers to say, hey, yeah, I think I can make a buck if I do that. Then until that time, I just kind of see folks doing the same stuff that they know how to do, what they've been doing because they did it, their dad did it, their grandpa did it, their great-granddad did it. But at least if we can encourage the conservation practice, that would be a plus. Yes, sir?
So speaking of making perennial crops profitable, what's the reason that on some of this marginal ground people aren't grazing versus, they're growing, corn or something like that?
I don't know. There's not a lot of money to be made out of perennial grass on your ground unless you're in CRP and getting a payment for it. So if you're not in CRP and you're not getting a payment, most of the farmers I've ever talked to, it really irks them to have ground that's not working for them, which means if they have to break it out of grass, if it was in previously CRP, just so they can work it and put row crops in it, that's what they'll do. Again, the reasoning why people make the decision that they do are myriad. I can't read their minds, but I think it's either a cultural decision, something that does a socio-behavioral something or other, which I don't know crap about, or they just don't have, there's not an economic factor there that would convince them to not produce. Although I kind of wonder about that sometimes. Like, if you really penciled it out, if you go to this already marginal land and you put row crops in it, are you really getting any money off of it? other than you just can't stand to see it not working for you, I really don't know how profitable that effort actually is. Yes, sir.
You talked about the importance of using the equivalent mass approach instead of the equivalent depth approach for measuring soil density.
Yeah, it makes a big, yes.
So I'm wondering, did you find differences in bulk density of the soils as a result of management?
Besides tillage?
Yeah.
Um,
Or including tillage, what impact did tillage have on bulk density and therefore on the calculations that you
So normally the bulk density in our tilled soils, we try to wait till the soils are pretty well consolidated before we take samples even in a tilled system. We're not going to wait till right after they disc it and then grab soils. But nonetheless, in general, we tend to see that those bulk densities are a little bit lower in the tilled systems. When we look at the perennial systems, we didn't really see a whole lot of difference in bulk density between the perennial and the no-till corn systems. So in that sense, I guess it depends on where in the soil you look. If you've got a lot of root material in a perennial grass system and that roots material is crowding out the actual mineral soil, it gets more and more difficult to determine what the actual soil bulk density is because less and less of it is actually soil and more of it is just roots.
Well, as a soil scientist, I'm always asking myself that question, what is the definition of soil in the upper 3 inches of perennial grass?
Yeah, because sometimes there isn't any. Sometimes it's just rhizome. Right, or very little. Yeah, it's a conundrum that we're faced with too. And bulk density, you'd think it's a pretty static number, and it is so not a static number. Bulk density sucks. It's like, there's so much about it. It changes from what moisture content, who's doing the Giddings rig. I mean, it's just, there's so many, it's a tricky measurement. It's a tricky measurement because we usually use a core method to do that.
Just as an expert in the field, what percentage of the variability do you think we see in numbers comes from the fact that we can't measure things like bulk density, right? Or figure out how much soil we
Ooh, girl, I don't know.
I mean, like the error of measurements, I feel like a huge amount of it comes from this part of the process.
Yeah, and I don't really know how to quantify that. I guess our hope is that whatever error you're making on one plot, you're making it on all the plots, and so you have like that level of variation is kind of held constant. So any treatment effects that you have still get expressed. But yeah, I think there's so much variability with just person to person methodology. I don't know. If you figure it out, though, that'd make an awesome paper. Yes, sir.
Along the same lines of Michael's question, so maybe you said what type of soils you were measuring in, or are they sandy, or what was the texture?
They tend to be silty clay loams here, or silt loams. We do have subsoils that get very sandy on that marginal site, again, because the underlying riverine sands from the old Platte, or the Todd River Valley or something is what they called it. So in certain areas we'll see, you can see even by the end of the growing season, the corn at the top of the hill's dead, and there's a knob up there where there's a really sandy subsoil, so it does very poorly. And then you get down towards the bottom of the hill, the sand is there's less sand, there's a lot more fine material, the corn does great. So there's a lot of landscape differences, even on that small study footprint.
The rates of carbon change you see are quite different. They're higher than what we see. There are a lot of people in here that have long-term experiments with similar types of management practices, and we don't see changes in carbon 10, 15 years out.
Yeah.
You can't measure them. It's still too variable.
Yep. And I think that has a lot to do with you guys' soils tend to be a little bit more fertile, I think, than what we have even in Nebraska. So I think that when you start off with a soil that's already kind of high in carbon, you've got a lot more buffer room there to move around. So it's harder and harder to see those treatment effects. There are some places that we've worked out there in Nebraska where, yeah, 10 years, 15 years, not really enough to see it. Or if we do, like we had measured this 7 years out, and even then it was, we didn't really see what was anything. And then, 12 years out, okay, now we're starting to see something. 16 years out, okay, we're 16 years out. Now we're starting to see a few things happening. But like I said, stakeholders, not everybody has that kind of patience to wait 16 years. So if you're lucky enough, like we were, to have people with foresight set the study up that long ago, then we kind of have a head start in the game, but that's pretty lucky. Yes, ma'am.
Have you looked at any of the microbial fingerprint of the soils?
We have. There are some way smarter people than I did that do the whole soil microbiome stuff. I know enough about that to be dangerous, but I don't really understand all of it. We did do some phospholipid fatty acid analysis there where we do see differences in the living microbial community composition, at least maybe not on we can't do it on a species basis, but if we look at functional groups in the soil, we do see those shifting around with both crop type and nitrogen rate or residue removal rate.
So it's interesting, after doing some reading, and it seems that there's a subset of microbes that harbor single nitrogen genes or fixation genes, and there's other ones that harbor whole suites of them. So I'm wondering if some of these fluxes that you're seeing, if you're seeing activity stimulated in some of these different groups, whether they have just one of these genes related to nitrogen cycling or if it's a whole suite.
I hope to have an answer for you here pretty soon. So the same 9804 site that we had, we actually got some funding from the Joint Genome Institute from DOE, and we sent those samples out to do the whole metagenomics work on there. And so one of the things that we're interested in is the nitrogen cycling on site. So maybe I will have some actual answers for you on that once we get a chance to really delve into the data that was returned to us. Yeah, it's cool. It's nice having these long-term sites. A lot of people want to come out and work on them, and we're happy to say, yes, y'all come and do some work that we're not able to do, but we'll provide you whatever support that you need to do the work that will just add to the body of knowledge. Any other questions?
I have one more very specific question.
Okay.
So, if you could go back to a slide, there was, I think it was your last half of your talk. There was the cave and rock at 120 in. There was like a stair-step increase in nitrous oxide emissions every year.
Okay, I'm gonna cancel out of this so that I can. Okay, somebody help me. I'm gonna get out of here. Escape. So a stair-step increase in gas emissions?
Yeah, it was an N2O. There it is.
This one? Okay.
So what is it about that variety? And it seems like just at that rate that it seems like you're building up potential for N2O emissions.
Yes, and so this is that same cave and rock. That's the one that's been there since the beginning of the study. It's a very mature stand. And so we think there's a lot of internal cycling going on here because it's just been there forever and it's still very vigorous. So in certain years, the cave and rock, it really doesn't need this much nitrogen. And in fact, Rob is convinced that it doesn't really need this much nitrogen either. We think really the optimum nitrogen fertilizer rate for switchgrass may be 35 to 40. So even, a third lower than what we've been applying. But that may also change with the maturity of the stand. Early on when you're getting it established, you'd probably give it more. But as it ramps up its translocation processes, it needs less. 2015, I believe, was one of we've had a lot of wet springs. And so there's a number of different reasons why we might see greater fluxes in one year than another, but I honestly don't know how to pick that stuff apart, right? So that happened to be a high flux year for cave and rock. I'm sorry, did that answer your question?
I'm
Kind
still
of.
puzzled by it, but
Yeah.
maybe it's establishment age or something.
It could, I mean, it could.
Biomass measurements, the soil carbon numbers, and the nitrogen stuff all side by side.
Yeah.
I mean, that's what mom's report.
Yeah, and it didn't happen every year. It just happened that one year. And again, maybe it's the method we're using. We have static chamber measurements that we get out there only once a week, once every 10 days or so. We take them. Did we capture all of the peak emissions? Maybe, maybe not. Maybe we got lucky that year and we grabbed a really high peak emission that we missed all the other years. Having some eddy flux tower data to compare our static chamber stuff to will be really, really nice. And so we're in the process of doing that with a couple of other sites that we have.
Okay, I think we are already a little bit late, so if you have more questions, Dr. Jin will be around. You can talk to her. Let's hang over to you one more time.